All right, thanks, Jim, and thanks very much for the invitation to speak here. It's been a long day, but thank you for sticking it out till the end. I'm going to talk a little bit about some of the things Jim just mentioned and some of the things that we've been doing more recently at Stanford in this area. So I was telling my colleagues that I'm coming to talk here at the Personalized Medicine Center, and they were saying, well, you don't know anything about that, which is always very nice of them to start with that. But they said, we already do personalized medicine. You don't need any of that gene stuff. And they said, yeah, well, and then he related to me this, I should say, I'm sorry, before I go further, I have this disclosure. So he said, this is what we do in clinic. You see, someone comes in and he says, your blood pressure seems to be a bit high. And so your doctor goes, well, just, what do you think I should do? Take this medicine. And then the patient says, well, it doesn't seem to make any difference. I took my blood pressure, it didn't make any difference. He said, well, take this one instead. Well, and then you get a cough, because let's say it's an ACE inhibitor, and so now you have a cough. And then you say, oh, well, let's try this other one instead. And that's a little better, but it really hasn't helped, so you try another one, and then maybe that's bound to work. And this is a situation that's repeated 1,000,000 times. I do this myself in clinic. This is what personalized medicine is today. We take a bunch of things and we throw them at the wall until we see what works. And I think you're all here because at some level or another you engage in the idea that we can do better than this. And so we had a thought a few years ago, around 2009, when genomes were starting to become a thing. What if everybody's genome was available in their medical record? We thought at some point that's going to happen. A patient's going to come in and they're going to have their genome. We thought we better start thinking about that. And at the time I was talking to, as Jim mentioned, Steve Quake, a colleague of mine. He's on the stage here tomorrow. And he was telling me a little bit about his family history. He'd published his genome, of course, that summer, not in USA Today, but rather in Nature Biotechnology. And I was asking about his family history, and I said, do you have any family history of heart disease? And he said, well, no, not really. And then he thought for a minute, he said, well, then there's my uncle, and there's my father, and then myocardial infarction, a heart attack, and he went on for a few minutes talking about a family history of heart disease, shown here in orange on his family pedigree. This is Steve right here, and everything in orange is heart disease. He also mentioned that his cousin's son had fairly recently died suddenly. This is his cousin's son here, 19 years of age, a completely normal 19-year-old, a black belt at karate who just failed to wake up one morning. A tragedy for his family, but also a challenge then for the idea that we could do better in terms of personalizing an approach to Steve and his future. And so I realized at that point, as we invited him to our clinic to come and be screened for heart disease because of all of this, that we were about to have the first patient walk into our center with a whole genome sequence. So when you have a problem like that, you call on your friends, and that's what we did. And in particular, my friends in this case were Russ Altman, who along with Terry Klein runs PharmGKB out of Stanford, and Atul Butte, who I think is also around at this meeting. And we put together a team and built some tools back in 2009 for the analysis of whole genome data. In particular, we built, I think, one of the earliest pipelines for Mendelian variant disease discovery, so looking at loss of function or open reading frame disruption across the genome. We built a genome-wide pharmacogenomic pipeline with PharmGKB and extending that to novel variants in genes, and a tools group built a predictive analytics for common variants of disease. And as an example of the sort of thing that could be done with that, we took these guidelines that are a little small for you to see just here. These are the cholesterol guidelines, which are kind of boring, but this is how we decide whether to give patients statins for cholesterol. If you run Steve through that algorithm, what you find at the end is that he runs right in the middle. And our question was this: if we took his genome and looked at the rare variation that suggested he had higher risk than average for his coronary artery disease, or the higher than even his family history would suggest, add that to the common variation that we have in his genome, add it to the drug response, which would suggest that he would have a positive response to a statin, and finally, the idea that he did not carry the variant that would predispose him to a side effect, so his genome suggested he would tolerate the medication well, then we get to a genomically informed decision. And I think we were lucky to be in a position to be able to make one of the first genomically informed decisions at that point in 2009. We found also at that point that people are kind of fascinated by genomes. We were lucky that there's a lot of interest from the press. I like to show this because if you look up here, you can see anything now with the data Andrew was talking about just earlier. This is there were 300 stories on the Google News. We're right next to Google. And you can see also the timeline of the interest in our celebrity, which started at 9 a.m. in the morning, peaking up here, and by 4 p.m. nobody cared anymore. So for those of us who think that we're celebrities, fame is very short-lived in this world. So, and thanks to Eric Green, who was on stage, of course, earlier, and his team at the NHGRI. It's also featured in this current exhibition, which I think is very good, at the Smithsonian Museum. Well, we went on from that to think about families. Again, Lee Hood, as well as many others, have said genome analysis is best done in families. And so this is one particular family who contacted us after we published that paper. John West, shown here, was the CEO of Solexa. At the time it was sold to Illumina, and technology, of course, is still the centerpiece of their product. And he'd been one of the first families. There were two at the time. One was sequenced on the Complete Genomics platform and one on the Illumina platform. And John contacted us to say, could we develop some tools, or what could we do to tell him about his genome? Well, we did the same thing. We started with a family history, and this is his family pedigree shown here. In particular, he was interested in the fact he'd had a pulmonary embolism, a PE, in his lung. And so that's not good, of course. And then he got treatment, which was warfarin. And then he had another PE. So he's in an unusual situation where he'd managed to have two pulmonary emboli, one on the actual approved treatment, in this case, the anticoagulant warfarin. So he wanted to know, could we tell him something about how that came about? And much more to the point, could we tell him something about whether his children were at risk for these conditions? And so we set out in a paper published here in PLOS Genetics to develop tools to extend what we'd done in Steve Quake's genome to a family of 4. In particular, one of the first things we realized was that there are issues with short-read sequencing, and particularly assembly using short-read, in that you're beholden to the reference sequence. And this is something that came up earlier on the platform today. In particular, the reference sequence, you may or may not know, has the risk allele for factor V Leiden, one of the most common causes of thrombophilia, the predisposition to clotting, which means that if you're homozygous for that reference and you're only looking at the variant call file, then you will not be found, despite the fact you might have somewhere between a 10 and 50x risk for having a clot. So we realized at this time and developed some new ethnicity-specific reference sequences where we substituted the risk allele for the most common allele at that position in the individual ethnicities, and used the ethnicity-specific major allele reference sequence for calling in this family. We did inheritance state analysis after the group, again in Seattle, David Gallis and Lee Hood, but extended it by plugging it into our interpretation algorithm to look across the family of 4 at genomic risk for multiple diseases. We also showed, using this hidden Markov model, as the group earlier had done, that the error rate was significantly reduced if you focused on the less complex areas of the genome. Well, we can do things like this. This is from a tools group again. We can show the whole family. This is the risk for type 2 diabetes based on 8 single nucleotide polymorphisms. There's many more now, but at the time we were confident in these 8. And you can see for the population, this is a Caucasian population in the background, the frequency spectrum for the occurrence of these SNPs. The x-axis is a likelihood ratio. And we have the parents here in the middle, right next to each other. But what you see is that the daughter's risk, shown in red, went up as a result of her distribution of alleles, whereas the son's risk in this family went down. And so if you can do this for one condition, you can do it for 6, you can do it for 8, you can do it for all the conditions for which we have data across the genome. And it's certainly for common variants, it's a very tractable issue to make these estimates. But we were really interested in his case in what might be causing the thrombophilia, the pulmonary embolism. And so we plugged in an early version of the phenotype mapping approach, where we take the condition of interest and we map it to genes that would be of interest from public databases again. And we came up with these. The factor V Leiden is shown there. But also these other 2 variants, in particular this Marburg variant, had some excellent data behind it. And this methylene tetrahydrofolate reductase variant also associated with thrombophilia. And in particular for John's question, did he pass these on to his kids? Then F for father and D for daughter, he passed both the factor V Leiden and the methylene tetrahydrofolate reductase variant onto his daughter. But we were also able to show that this variant here was passed on from mother to daughter. And so Anne West, in this case, John's daughter, was one of the authors on the paper, you might have noticed at the beginning, because she had worked in George Church's lab over the summer and had actually been analyzing her own genome. In fact, that was her AP science project, was to analyze her family's genomes, which is possibly a bit scary if you're her science teacher. But this is how far we went, and we were able to give her advice that she takes to heart in living her life. There are things that you can do to reduce your risk of having a clot in the lung, especially on long car journeys and other things. So this was some of the earlier days. This morphed into the more recent project, which I'm going to tell you with some unpublished data now. This was a project we started in 2011, just finished up at the end of 2013, where we took 12 individuals and sequenced them completely on Illumina and Complete Genomics platforms and ran a complete analysis on it all. It has become important, particularly in the last year, because of the profile given to the incidentalome after the ACMG guideline document, to think very broadly. Although much of the genome sequencing we do is targeted towards a given disease, can you find the answer to this mystery syndrome, for example? The incidentalome, what you get, other than with the focused look, is becoming very important, and whether we should disclose that back to the patients is a topic that is recurrent. So what we know, and what we know is that it can be challenging to do this. And these are some of the major challenges here. The genome is complex, that's not news. But a large amount of it is repetitive. Maybe even as much as 2/3 of the genome is repeat sequence. And there's also large areas of homology. If you're using short-read assembly approaches, then regions of homology are challenging. How do you know that that's the correct place for that short-read to go? Coverage is highly variable. I'll show you a couple of graphs in the next slides. We're also not very good at calling the type of genetic variation that is most impactful. So if we draw a graph of the average medical importance of the class of genetic variation and how good we are at calling it, the line goes like that. So we're very good at the not very important variation, the sort of single nucleotide variation that tends to lead to very low odds ratios. But we're actually pretty bad at the stuff, large rearrangements or even small indels that disrupt the frame. We're really not very good at that, and I'll show you some data to back that up. So what about the coverage issue? First of all, this is a gene that will come up at the end of the talk. This is KCNH2, this is a potassium channel variant. And this is the adjuvant sure select. The coverage, depending on how it's defined, is in the sort of 70 to 100 range, overall median. But look across the gene. This is 60 cutoff. So you see these parts here go way up here. So the coverage in this region is very, very good. But there's other regions of this gene, for example here, where the coverage is 5 base pairs, four base pairs. It's covered down here below 20 again. And here, shown at the bottom, are variants that are known to have clinical medical significance. So these are not areas that are alternately spliced, that aren't important, that don't have functional relevance. These are areas of the genome where we know there are genetic variants that are of importance. You can fill these in using a variety of approaches. This is one that Personalis, one of the companies I'm associated with, does. You can see in green the filling in, but there are other approaches that other companies are taking. This is the dilated cardiomyopathy gene with a similar thing. It's better with whole genome sequencing, but it's not perfect. If where we were before was this top line, these are the 56 ACMG genes. And if we said that before we covered every base pair at quality with Sanger sequencing, then the line of standard for clinical grade genomics should be 100%. As you can see in red for Illumina and blue for complete genomics, we are significantly below the line on very many of the genes that we are now suggesting we give back incidental findings to our patients. Also, the concordance for single nucleotide variants, shown here in gray, is very high, but for indels, shown here, is much lower. 55% concordance for indel calling between the two platforms. So we have some work to do. What do you find in a genome? Well, you find about 100 genetic variants. It takes about an hour to do the manual labor in terms of looking and assessing the evidence for that. And there are probably 1 to 3 initial diagnostic tests costing a few $100s. We reclassified 70% of the variants downwards. But there's also great hope here. I'm telling you about the limitations because I think we have to be practical if we're going to realize the power of this technology. We found in one particular patient a BRCA1 variant. There was a founder mutation that had a very clear evidence base, and that patient was able to take medical action to prevent future mishealth. And of course, this has been a big story in the last year, and many of our clinics, Jim's clinic and many others, have been overloaded since this story came out. So Amir, at first thing this morning, mentioned the Clinical Genomics Service that we're launching at Stanford. We're taking the work that we've done, and I've shared with you over the last 10 minutes or so, and bringing it to the bedside of the patient, starting with a whole genome germline, and moving shortly after that towards tumor sequencing and pharmacogenomics. And then I'll finish with the story that was actually the title. Originally, this talk was going to be 5 minutes, and I was just going to come up and tell you a story about a baby that we sequenced recently in the neonatal intensive care unit. So here's the promise of the title of the talk here. Long QT syndrome is a syndrome associated with sudden death. The ECG is what you get when you take an electrical tracing of the heart, and you get these waves that bizarrely are called for the middle of the alphabet, not the beginning. It could have been A, B, C, D, E, but it's P, Q, R, S, and T. And Q to T is the part where the heart resets every beat. So if the QT is too long, you're at risk for an arrhythmia that could cause sudden death. And this is unusual as a syndrome in that it's now defined by its genetics. It was previously defined by whether you had deafness or not, and now it's defined by its genetics. So this is a little baby that was born electively 2 weeks early by C-section because she was noted to have bradycardia, which is a low heart rate. And she had bradycardia not because her heart had heart block, because the electrical signals weren't going down, but rather because the QT interval, the resetting interval, was so long that there wasn't time, that the next beat sort of came in the middle of that. What we did was, with the help of the Illumina CLIA lab, on day 3 of life, which is essentially still 2 weeks early, was sent the rapid turnaround CLIA whole genome. And by day 5 of life, we had identified the variant that caused the long QT syndrome. So this is not a mystery disease. We knew what the disease was, but we didn't know the cause. And in this case, knowing the cause reclassifies the condition and helps change therapy. Previously, when we did this, this would take 6 weeks or longer. It would cost $4,000. You get 8 genes. And in today's world, we get all of the genes for $12,000. We're good for single nucleotide variants. We're less good for the other classes of variation. So I'm going to wrap up now, but in conclusion, I just wanted to say personalized medicine is here. The tools are evolving. There is a significant potential, I don't need to tell you this, for cost-effective discovery and real change in people's lives. But we really have to demand a clinical grade quality. We have been used to demanding 100% coverage and 100% quality. And we've moved to a world where we've moved to next generation sequencing, and we're accepting 90% coverage at 10x or more. And so I would urge you to ask your providers and ask the people that you're ordering these tests from for clinical grade quality. And especially if we're thinking about disclosing incidental findings in genes that are not well covered, I think we have to be very careful what we know and what we don't know. So great potential, but some realism as well. These are the people who did all the work, of course, and the people who gave us the money to do some of it. And special thanks to both Personalis and the Illumina CLIA lab for turning things around and working very long hours at the weekend for these little babies. Thank you so much.